Continuing chapter 5, section 1. This is, you probably can read this, but just in case. First panel, hey, what's up? No, you don't. Why? You don't want to work, so you're prowling for victims to procrastinate with. Well, you're not sucking me into your vortex of unproductivity. I have work to do. I hear there's new candy at the vending machine. Darn you, let's go. I don't know if it's easier to put stuff off when most stuff is remote and you're doing everything at home and there's, maybe that's a way to get yourself distracted from just being at home all the time, except for being in lab. But I know it's a weird semester, and I know no matter what the semester is, it's always difficult to keep yourself focused and moving forward. Is setting yourself up a schedule is not a bad idea. We're moving into proteins. Proteins are a polymer, a long string of amino acids. Amino acids have an amino or an amine group on one side and a carboxyl or an acid group on the other side. There's NH2, there's COOH. It's typically virtually always got one H on the central carbon and then what they call an R group that isn't labeled up here as an R group, but it's an R group. And these vary quite a bit. Sometimes they're hydrophobic, sometimes they're hydrophilic. You can build a protein with a hydrophobic layer and then it inserts right into a cell membrane that also has a hydrophobic layer. All sorts of neat tricks that you can use by stringing these things together. So there are roughly 21 amino acids that you find strung together in proteins and living things. Obviously, there's way more than that. You could make almost an infinite number of amino acids. I'm sure chemists could probably come up with a number. But the R groups could be a lot of different things, but they aren't. There's only about 20 of them. I think you have to really dig to find the 21st. Here's a string of them. They connect carboxyl group to amino group, carboxyl group to amino group. Now they haven't put the bond, they put the aminos here, here's the bond. And again, it's dehydration synthesis. What you've got here is water coming out, and instead of an oxygen bridge, you're connecting the carbons to the next nitrogens, called a peptide bond. They used to call amino acids peptides. Memorize these. No. In some advanced classes, you would need to memorize all the different amino acids. But you'll notice that these are the R groups up on top. They can be pretty much not there, just an H. They can be just a methyl group. They can make weird rings and connect back to the nitrogen. You've got a propyl group there. Sometimes you get a benzene ring. There's sulfur. Here's another nitrogen. Here's another nitrogen on the end of a string. So here's another sulfur. So what's on the R groups do vary quite a bit. What also happens is you have an amino acid and you're making a string. That N to C bond looks like they're all lined up like this. But because of the overall electron cloud imposed by the R group, is that the bond between A and B amino acids may be like this, between B and C may be like this, between A and D might be like that. So as you go down the string, the amino acids connect in a way that three-dimensionally keeps the string from being perfectly straight, and in fact can curl the strings back around and bring one string going past another string and then cross-connecting, sometimes through sulfur bridges, sometimes through a lot of times through hydrogen bonds. But this is basically how a protein is constructed. The way that these connect also, the chirality of them all takes the same twist. There's what they call a left-handed twist versus a right-handed twist. And one thing that's always been a mystery is that all amino acids, and when I say all, you should always see that in quotes, all amino acids in living things have the same chirality, they have the same twist. And that's an interesting thing people try to explain. There's a couple of links in the book to potential explanations for why. Because if you were to whip them up in a test tube, you'd get a 50-50 distribution of left twist, right twist. Another picture of the bond. Now, proteins are complex molecules, and as such, they have different levels of structure. Four, well, sort of four. I'll come back and write some of this down, but I'm going to move this. You have primary, secondary, tertiary, and a lot of times quaternary, but not always quaternary. That's why it's not always four. This is primary structure. What's the order of the amino acids in the string? Each of these little boxes represents an amino acid. So you've got aspartate and cysteine and threonine and alanine and glycine and leucine. There's all sorts of different amino acids just in the string. Not represented here with any, they just want to fit it all on here and look kind of artistic. And this doesn't necessarily represent the way that the bonding angles would clip up. But the way the bonding angles clip up becomes an important level of structure inside a protein, and that's secondary. Sorry about the sneeze. Excuse me. You get repeating patterns. Up, down, up, down, up, down, up, down. And very often, you'll get a couple of strings going past each other that are also going up, down, up, down, up, down, like you get here and here. They call them pleated sheets. That is inside the protein, but it is an internal pattern. And sometimes you get spirals, what they call a helix pattern. Those are your two major secondary structures inside this. Now well, we'll see this on another one. You can see here, they're trying to represent the overall outer electron cloud. of this. This outer electron cloud is tertiary structure. It is the overall shape of the molecule. Let me try to find what I'm looking for. There we go. Now what you're looking at is sometimes tertiary structure and sometimes quaternary structure. You can't tell from just looking at the outer electron cloud here, but they're showing you the shapes of the molecules. In this case, they're showing it connecting to a DNA molecule. In this case, they're showing it connecting to an inhibitor. But you've got a bunch of proteins here. Some of these are enzymes. How do I know that? They have an ASE ending. And that reminds me of something I left out of your notes earlier, that you're going to want to go back and write this in. Carbohydrates, pretty much, except for really old ones that were named before the rule came up, carbohydrates have OSE endings. Glucose, sucrose, cellulose. Is that OSE is an indicator that you're dealing with a carbohydrate. The ASE ending is an indication that you're dealing with an enzyme. And we'll put enzymes on the list later on, but the ending, I gotta put that, I don't think that was even in my notes, that's bad. And this is glutamate synthetase, alcohol dehydrogenase, phosphofructokinase. A kinase is an activation enzyme. Lots and lots of enzymes here. Scorpion neurotoxin, glucagon, and insulin. Insulin causes liver and muscle and fat cells to sequester glucose. The fat cells convert the energy, use the energy to make lipids, but it sequesters the energy. And glucagon gets sugar release from your liver cells. It says, give us some of that sugar back. You stored it as a starch, we want it back as a sugar. Interleukin B is an immune system molecule. This is the basic shape of an antibody, where these ends would attach to antigens, to foreign molecules, and this would be an activation end. And remember, this is all shape. This attaches, it changes the shape of this, and it goes from being just an antibody floating around to basically a signal to the immune system that says, whatever we're attached to here, go after it. So, we've got primary structure, leave yourself some room. Secondary, tertiary, and not quart, quaternary. Wait a minute. I knew something didn't look right. Quaternary structure. First, second, third, fourth. String of amino acids. Internal patterns. Overall shape. Now this fourth one, some functional proteins are more than one string. They're more than one lump combined to make a functional lump. In that case, quaternary is how do the lumps fit together to give you the functional protein. Hemoglobin is two pairs of lumps fit together to make the hemoglobin molecule. And each one of those has a heme group, has an iron group in it as well. So something like insulin, I'm pretty sure, Insulin just has tertiary structure, just one string. Hemoglobin has quaternary structure because the overall lump is more than one lump combined together. So you always get this, but you only sometimes get this. So string, internal structures, internal patterns, overall shape, how lumps fit together to give an overall shape if there's more than one lump. In the primary structure, each amino acid, we'll come up to this later, is coded by a three nucleotide sequence in your DNA. It's a 3-to-1 code. So the sequence of DNA gives you the sequence of amino acids. And we'll go through those codes later. And we'll also talk about it when we get back to DNA. The string starts at the N end and goes to the CN, what they call the amino terminus to the carboxyl terminus. I mentioned what gives you your overall structure is sometimes the strings are covalently bonded from one string to another to hold it in position. Those tend to be much more stable proteins. Sometimes it's hydrogen bonding. And the strength, remember the strengths of hydrogen bonds can really vary depending on the unequal sharing of electrons. And also in this case, sometimes the distance is that the two strings can be hydrogen bonded, but the other constraints keep it from being too close or too strong. Anything that can disrupt the internal bonding can cause the protein to lose shape and lose function. That goes to, they gave that an old word when they started to mess with proteins and they stopped doing what they were supposed to do, and they called that denaturation. They lose their nature when you heat them up or you expose them to hydrogen ions or you expose them to hydroxide ions. There's a whole bunch of different ways to make a protein shape disrupt. Now, some proteins, once you've disrupted their shape, like with albumin, with egg white proteins, it tends to interact with the unwound proteins near it and lock together. But a lot of proteins, once you have kind of denatured them, if you change the situation back to what they were in before, they'll go back into the same shape. They'll call that renaturation. Remember, that's why we have acid in our stomachs and one little enzyme that clips the strings in particular spots. Therefore, you've broken the primary structure. You're not going to get a reconnection of tertiary structure. Also, inside these molecules, when I talked about the antibodies, this one connects to the antigen, this one is a signal to your immune system. Areas within a protein that do different things are called domains. Proteins embedded in the membrane have hydrophobic domains. The heme group is called the prosthetic group. I'm going to write some of this stuff down. Grab my notes so I don't forget any of it this time. Checking the time. Prosthetic groups are not always minerals, but they often are. Then you have complexes, which is pretty much what it sounds like. A lot of functional bits in a cell are a bunch of proteins interacting with one another. They're not all quaternary structure. They're not like all combined bits into one protein, but something like a ribosome, which takes your RNA and uses it to make protein strings, to make amino acid strings. That is a complex. A chromosome is a complex. In that case, it's a complex of protein and DNA. So a complex is just a combination in a particular spot doing usually a fairly complex job as well. When a protein is being made, it's being made amino acid by amino acid, so it's being built on primary structure. As the next amino acid comes in and connects, you get these changes in bond angles. So it starts to come out with at least some of the inbuilt secondary structure. But there's a whole bunch of different ways that most proteins can go as they're folding up. And they need to be directed into particular forms. There are other types of proteins waiting for them as they come out of the building, the ribosomes where they're built, that make sure they get folded into the right conformation. Those are called chaperonins. And similar to we were talking about prions, how a prion can grab a protein that matches it by primary structure, but not by tertiary structure. And it can make that protein become a prion by changing the tertiary structure into a prion structure. And then it can go on and do the same thing to whatever functional protein matches it inside a cell. Mad cow disease is a prion, is that the prions are going after a functional protein in your nervous system, primarily your brain, and changing those proteins into prions. The more of them that get changed, the more of them that can change more, and the fewer functional proteins are doing their functional protein job in your brain. And your brain goes bad. Some of them work very quickly. It's classic Creutzfeldt-Jakob disease, works very slowly. But mad cow disease works within a couple of weeks. Creutzfeldt-Jakob within decades. But I'm just gonna mention, I'm not gonna put it here. We'll talk about it later. Okay, there's a lot to talk about with proteins. This is as much to fit on this particular video, so we'll stop here.